Molecular genetics studies the structure and function of genes at a molecular level. The study of chromosomes and gene expression of an organism can give insight into heredity, genetic variation, and mutation. This is useful in studying developmental biology and understanding how to treat genetic diseases.
Through molecular genetics technology, scientists can study diseases and the role that the specific genes play in an organism by using transgenic animals. These are animals that have foreign genes deliberately inserted into their genome.
Most commonly used transgenic animal is a mouse. A genetically modified mouse has its genome altered through the use of genetic engineering techniques. One such method is to selectively produce transgenic mice using green fluorescent protein as a marker. The photo shows two transgenic mice positioned on both sides of the plain mouse. These transgenic mice have been genetically modified so that they carry a green fluorescent protein which glows green under the blue light.
In molecular genetics, the DNA has a series of functions that are explored in the laboratories by scientists. One of the most basic techniques is the extraction of DNA from a cell. This DNA can be later used in a series of other experiments. This extracted DNA can also be fragmented into smaller pieces by restriction enzymes when a more specific part of the DNA is wanted. These fragments are often spread out on a gel by its size, and one can identify these fragments with the help of a molecular probe. Amazing things are done with these fragments, such as cloning a gene or any other fragment of DNA. The final product is a transgenic organism.
One genetic technique you can do with DNA in the lab is to extract it. DNA can be extracted from many types of cells. The first step is to lyse or break open the cell. After the cells have been broken, a solution of sodium chloride and detergent is added. This breaks down the cell membrane. After this, ethanol is added because DNA is soluble in water and causes the DNA to precipitate. Lastly, the DNA is isolated and washed.
And this was a little introduction of some molecular genetic techniques you can do with DNA.